Well, good evening, ladies and gentlemen, members and friends of the RSC Belgium section, to our final lecture before the summer break. It already looks like summer around here. The temperature is getting a little bit warmer. This evening we're very, very pleased to welcome as our speaker Pierre Sonveaux from the Universite catholique de Louvain. Pierre is a pharmacist by training. After doing his doctoral thesis in vascular oncology, he graduated in 2004 and then went on a postdoc to the lab of Professor Pierre in Duke University, North Carolina, USA. And when he came back to the University of Louvain, he received an ERC starting grant and was promoted to assistant professor, and then he created his team to work on translational research. Now he has a research group of some 15 researchers, currently active in 3 different areas: the understanding and transformation of drugs in the oxidative pathway of lactate in cancer, the characterization and harnessing of metabolic control pathways in cancer metastases, and unraveling the contribution of cancer and host cell metabolism to the resistance, both chemio and radio, for cancers. This evening he's going to talk to us about towards the prevention of local recurrence and distant metastases in breast cancer. And as you will hear, he has some very exciting new results to present us, which I think you will find extremely interesting. Pierre, it's a great pleasure once again. And the word is with you. Could I ask our members and participants to cut their camera and their microphones? At the end of the talk, you'll be able to come back in as you have questions. I just remind you that the next lecture of the RSC will be on the 29th of September, when Vincent Lemaitre will give us a conference on recent advances in high energy physics. Pierre, we look forward to your talk. Thank you very much for coming this evening.
Well, thank you. First of all, thank you for the invitation and the nice introduction. It's a real honor for me to be invited by the Royal Society of Chemistry and the Belgian branch in particular. And so, as you said, today we will speak about cancer and cancer metastasis in particular. And I will make an update of a new therapeutic strategy that we have for the prevention of recurrence and distant metastasis. I think that an easy starting point for such a presentation of on cancer is the description of the tumor growth agenda in the body. And if we look at the initial steps of cancer growth, well, you have an initial cell that undergoes damage to DNA, which will activate oncogenes and suppress tumor suppressor genes. And a combination of these mutations will induce the cancer phenotype, which means essentially that this cell will give rise to a progeny of cells that will have their own agenda, and the agenda is to colonize the body. Initially, a microscopic tumor will be formed around blood vessels, because as you know, in our body, the network of blood vessels is very dense. But then the tumor will grow at an increasing distance from the existing blood vessels, and the network will enlarge, and some tumor cells will be confronted quite rapidly to the establishment of a gradient of hypoxia, so a gradient of oxygen from the tumor supplier, so the blood vessels that are supplying oxygen towards the periphery of the tumor. And so now in dormant tumors, you have 2 populations of cancer cells. You have those that are correctly oxygenated, and so they can behave as a regular cancer cell and proliferate. And then you have those cells that are in a low oxygen environment, and the oxygen level is not enough to ensure all the cell functions. And these cells, if they are not adapted to hypoxia, they will just die. So in a dormant tumor, you have a dynamic equilibrium between the proliferation of oxygenated/oxidative cancer cells and the death of hypoxic cancer cells. And this can stay in the body for years. In most of the cases, these small tumors that are not that are not diagnosed because they are too small and are not symptomatic. In most of the cases, these tumors will regress spontaneously, especially because immunity will take care of the problem. Just to illustrate this, this is a slide of a tumor mass in a mouse, so it's a human cancer. And this is a picture that I took when I was a PhD student, just to show you that this is the reality. You have the tumor blood vessel here at the center, and then you have a number of cancer cells that are growing at an increasing distance from this blood vessel. And if you go too far, you have around the living cancer area, you have cells that are dying through necrosis. And this is because of the gradient of oxygen that establishes between, from the vessel towards the periphery. And it is simply due to the consumption of oxygen by the layers of cancer cells. So the faster the longer the distance, the less the oxygen. And this is also true for nutrients. So the farther the distance, then the less the nutrients. So I was told that the public is not a public of cancerologists or oncologists, and so that I had to simplify a little bit the talk. And well, this is a scheme that I like to show to the students to explain the situation. It's an easy illustration, and it's mostly anthropomorphic. So here is the cancer cell, and this cancer cell is facing two problems. First one is a low level of oxygen, hypoxia, and the second one, as I just told, is a low level of nutrients, and here is glucose, and there is a low availability of glucose in tumor areas that are at a distance from these blood vessels. And then because also the perfusion is low, well, because of that, wastes will accumulate. And the main wastes that you can find from cancer metabolism are carbonic acid, of course, when the cells are oxidative, and lactic acid when the cells are fermentative. In addition, if we look a little bit further in the history of the cancer, if this cancer evolves, then it will be attacked by us, by the doctors. Attacked by what? By chemotherapy, by immune therapy, and maybe by radiotherapy as well. And this will create, all of these will create a selection pressure on the cancer cells. And so the cancer cells, when they are in a hostile environment, and when they are attacked, they will try, of course, to adapt and to evolve. If I replace the cancer cell by an individual, let's say this guy here is a survivor in a plane crash. So a plane crashed in the desert. There are few surviving people. Here is one of them. Of course, there is little water in the desert, little food. The site can be attacked because maybe they're in a country at war, in Libya, or for example. Yeah, there are only few survivors. And so it's a question that I asked to the students in the frame of adaptation and evolution, how could one survive in such a metabolic desert? And you can rephrase, in such a metabolic desert, how can a small number of cancer cells, how can they survive? And there are many possibilities to think about. The first one, the most logical, is when the plane crashes, you try to find food in the remnants of the plates, of the plane. And so you will find some types of food maybe that you don't like to eat usually, but you will eat it because this is the only food available. Even for example, if you have to eat paper because there is only paper left. So the first adaptation is metabolic plasticity. And this is typical of cancer cell. When a specific resource is lacking, then they will switch to another resource. So for example, when glucose is lacking, they can switch to lipids as a food, as a fuel, or they can switch to glutamine or proteins or whatever is available. Then you have different strategies between the single individuals. So if I have water and you have food, we would live longer if we exchange. So I give you a little bit of water, you give me a little bit of food, and we will both live longer. So these people can cooperate. Cancer cells can cooperate. And this is one of the topic of the studies in the lab, a cooperation based on the exchange between cancer cells, of the exchange between lactates and glucose. I will not talk about that today. The opposite is to enslave. So you can enslave the other cell, and you can instruct her to work for you. And so you will start to work for me. You will give me all the food you have, all the water you have. You will die fast, but I will survive longer. This is another adaptive or evolutive strategy, as you want. Then if you think on all the possibilities, you can eat your neighbor. This is cannibalism. Cancer cells do perform cannibalism. They can eat host cells or they can eat other cancer cells to survive longer. You can try to find a radio in the plane and to call for help. And then maybe an helicopter will come to save you, to rescue you with also food and water. And so cancer cells can produce and secrete factors that will reach distant blood vessels, and this will be really a SOS code. Help me, I need more blood here. And the blood vessels will start to grow and to reach the areas that were deprived. This is called angiogenesis, from the Greek angio, blood vessel, genesis, formation. If you think about all the different strategies, one of them is hibernation. If I dig a hole and then I wait long enough in the hole, maybe the desert will be transformed in a forest after 10,000 years. Cancer cells can do that. And these cancer cells are called stem cells. They can stay quiescent for years, months, years. And then when the situation is better, they regrow a cancer. They are typically those that are responsible for cancer recurrence or relapse. And then one of the possibilities would be to leave the area. So you take your backpack and you go through the desert to find an oasis where maybe the grass is greener, you have more food, you have more resources. And this is typically what metastatic cancer cells do. The success rate of finding an oasis is low. So a lot of cells will die. A lot of these people here will die, but maybe one of them or a few of them will find a final destination. And so this is summarized in the slide here. You have metabolic plasticity, cooperation, enslavement, angiogenesis, cachexia. So it means that the people with cancer at advanced stages, they will lose weight, muscular weight and fat weight. Why? Just because the cancer is mobilizing all the resources of the body at a systemic level. And so the cancer wants to compete with the body to eat what is available. Cannibalism is illustrated here, where a cell can engulf another cell and digest it in the autophagic particles. This is autophagy. So the cells can eat itself, and so it can eat, for example, the proteins and the organelles that it doesn't need in a given situation. For example, if there is very little oxygen, do you really need mitochondria? Maybe the answer is not so much. And so they will maybe tend to digest their own mitochondria to produce energy through another way, through fermentation. And here is illustrated the hibernation. So you have a cancer that is treated, few cells will remain, the tumor will regress, so there is a positive response in the clinics. And if the cancer is not absent for at least 5 years, then it can regrow at year number 2, 3, 4, whatever, and you have a recurrence or a metastasis that will develop. And so today, as was introduced, we will focus on metastasis. And as you know, metastasis is one of the main reasons of failure of cancer treatment in the clinics. When a cancer is very localized, you have many different strategies that can be used. One of them is surgery, another is radiotherapy, so that is targeted on the tumor area. But when you have 1, 2, 7 metastases that are present in the body, then usually patients go to palliative care. And so then the intention is not to cure the patient, but the intention is rather to have the best quality of life for the remaining duration of the life. So we wanted to do something in this context, and we wanted to study metastasis. And so I just want to present you the next part of the agenda of tumor progression in the body. And this is also the working hypothesis of the lab. And so we believe that those tumors that escape the immune attack will enter in exponential growth because there will be 3 main adaptations that we can order on a timeline. We believe that the first adaptation is a metabolic adaptation, which we call the glycolytic switch. And it means that the cells that are no green here, they have modified their metabolism. So the cells, the original cancer cells, they were oxidative, so consuming glucose, lipids, or other nutrients and through respiration to produce energy. And now these green cells here that are represented, they are fermentative. And so they will convert glucose to pyruvate and then pyruvate to lactate, and lactate will be excreted. This is lactic fermentation. And in this mode, you produce low levels of ATP. So the rate is low. So 1 molecule of glucose consumed gives 2 molecules of ATP produced, compared to 36 to 38 through respiration. But these cells can also accelerate the glycolytic rate. So by unit of time, they will consume much more glucose compared to the oxidative cancer cells. And in the end, they are able to produce similar levels of ATP compared to the oxidative cancer cells. And they dialogue at the biochemical level, as I told you. They can exchange resources. And as you understand, oxidative cancer cells can spare glucose, so that the glucose will be available at high levels for the fermentative cells, that in turn will fuel the oxidative cells with lactate as an alternate oxidative substrate. So first adaptation, metabolic adaptation, which will allow the cancer cells that are hypoxic to survive, to proliferate, and to invade the tissue. Second adaptation, the angiogenic switch. New blood vessels will grow out from pre-existing blood vessels, and they will also deliver oxygen and nutrients to remote tumor areas. This will further promote tumor growth. And then you have the metastatic switch, where some tumor cells will be selected to leave the primary tumor, enter into the bloodstream, travel through the bloodstream, and they will stop in a distant organ, such as here the lungs, for example. But it can be other organs such as the liver or the brain, or it depends on the type of cancer. And then there they will establish a new cancer, a new tumor, which we call a metastasis or secondary tumor. And to do that, the cells have to possess simultaneously a lot of properties. The first one is to be able to detach from the neighbors. And this is usually it triggers cell death, but cells can be adapted to resist to this kind of death. They have to migrate, and they have to migrate directionally from A to B, B being the blood vessels. They have to invade, because you can imagine that there is collagen and a lot of deposition in the tissue, so they have to digest the tissue. Then they have to intravasate, so they need deformability. They will be reoxygenated in the bloodstream, so they need to resist to redox aggressions. In the blood, they will encounter immune cells. They have to be protected against immunity. And then when they arrive in the distant organ, they have to know they are there, which is a question that we are digging for the moment. There is no explanation for the moment of why a cancer cell stops in a given organ for a metastasis and not another type of cancer. And then on there, in the destination organ, the cell has to extravasate and to be able to repopulate, to remake a full cancer. So they need stemless properties. So stem cell is a stem that is multipotent and has the capacity to regenerate a tissue from a single cell or a very small group of cells. Well, so you have cancer stem cells that are able to regenerate a full cancer from a single cell or a small group. So if the hypothesis is true, and if the timeline is true, it means that all the metabolic adaptations that can occur at the beginning here will influence and will probably promote the rest. So it will probably promote angiogenesis. It will probably promote metastasis. So the key question that we address is there a metabolic control of tumor metastasis? Because if this is the case, then we can target metabolism to prevent metastasis. Well, we will now enter into the result area, so it will be a little bit more complex. I hope that you have all understand the context. But I will take just another image to illustrate how difficult was the approach for us and for the community in general, because in an advanced cancer, so in a patient with an advanced cancer, it is estimated that about 1 million of cancer cells get access to the blood per gram of tumor per day. This is a very high level, a high number of cancer cells that have access to the blood. And among these, only very, very few are able to make a metastasis, because only very, very few possess all the characteristics that I told you. And these few are called metastatic progenitor cells. And so they represent 0.00001% of the total population of cancer cells that are circulating in the blood. So it means that the material to analyze is not available in high quantities. If I take an illustration, I would take the Grand Place of Brussels because we're in Belgium. So if you have a crowded Grand Place of Brussels and there is a terrorist on the Grand Place and the police has to identify who is the terrorist to inactivate him or her, well, it's difficult because he's only one or she's only one. So how to know it's a terrorist? But if on the same Grand Place of Brussels, you have 100 terrorists, all the same, that are on the Grand Place, it's much easier for the police to identify them because they're all the same. So a strategy to be able to identify the terrorists is counterintuitive, but it would be to increase their numbers and to hope that they have the same uniform. This is what we did. This is a strategy that we used in cancer. So these metastatic progenitor cells are very few. And so we decided to make a situation that is a bad situation because it's a metastatic cancer in a mouse model. So we decided to make it worse and to render the cancer more metastatic. So we had more material to work with. And during the selection process, we could then compare the initial cells that were indolent or innocent to the terrorists, so to the metastatic progenitor cells, and try to find differences, because any difference would make for us a potential strategy to inactivate these cells. So this is the philosophy, and now this is the realization of the philosophy. How did we do that? Well, in vitro, it's possible to select cells for one of the characteristics of metastasis, which is invasion. And so in that case here, we have human cancer cells that are called CIHA. They are from a cervix cancer. And these cells are put in a top well in a transwell system. So you have two wells, a top well, a bottom well. They are separated by a porous membrane, and the membrane is covered by a matrix similar to collagen. And so if the cell is invasive, it has to dig its way through the collagen and then to cross the porous membrane and to reach the bottom. And to ease the situation, the cells were labeled with fluorescence, so we could easily pick them. And so the idea was, if you do that, so in this system, you take the few cells that made it overnight, and you make a second selection. Maybe more cells will make it. And if you make a third selection, maybe even more cells will make it. And so this is pure Darwinism. If you select cells for a given property that gives them an advantage, the number of cells should increase. And this was the case, of course. Otherwise, I would not be here tonight. And I show you here that these invading cancer cells in vitro, they increased already after 3 rounds of selection, so which is quite fast. And the difference is an increase in 3-fold in invasivity, which is rather important. The migration can also be assessed in the same system. The only difference is that there is no collagen matrix here. And so this is simple migration from A to B, so from top to bottom. There is a chemoattractant. And here, as you can see, the migration was increased by about 5-fold. And then, as I told you, if we want to know why these cells became so aggressive, we can compare the daughter cells to the parental cells and make the difference. And so this is what we did. And as our focus was on metabolism, we measured the glucose consumption first. So you have the wild type cells compared to the super invasive cells. And as you can also see here, there is a 5-fold increase in glucose uptake. And when the wild types or the super invasive were put under hypoxia, they increased the glucose consumption, so to increase the rate of ATP production per unit of time, but to a similar level that was not different. So we try to understand here this difference. Where was going glucose? What was the final destination with the increased glucose that was uptaken? And the answer is by the negative. When we measured the lactate production, there was no change in the production of lactate. So it means that glucose was not fueling lactate fermentation, but rather it was fueling cell respiration. Intermediate conclusion with this model, the increase in the glucose uptake fuels cell respiration. And it means that the next hypothesis, of course, will be, is an oxidative metabolism or a highly oxidative metabolism, is it necessary for a metastatic progenitor cell to be such a metastatic progenitor cell? easy model, a bit more complex, but more clinically relevant, perhaps, is the same approach, but it was done directly in mice. So here we have a mouse melanoma model, which is called B16F10. It can be injected under the skin of a mouse, and then with time it will give formation rise to a few metastases in the lungs that can be pinpointed quite easily because they are black, they have melanin. If we microsurgically dissect these metastases, they can be put in culture and then injected to mouse number 2, where we expected to produce more metastases to the lungs, and so on and so forth for as many selections as you want. This is Darwinism, and we expected to have more metastases in the end, which was the case. And this was, so this is quantified here with a number of mice. So this is 4, so passage number 4, B, because we made several selections in parallel to be sure not to select peculiarities. These cells were tested in vitro for the invasion capacity. Invasion activity was increased, and they were also able to make more colonies in a dish. So if you simply compare the B16F10 here to the M3 after 3 rounds of selection in the mouse, or M5 after 5 rounds of selection in the mouse, each dot is a colony. So each dot represents a stem cell that has generated a progeny. Okay. And so this is one of the main characteristics of metastasis. And so this activity was also increased. And so we had here another good model to compare the cells, let's say the super metastatic cells, to the poorly metastatic cells, the poorly metastatic parental cells. So on the left, you have 1 terrorist on the Grand Place of Brussels, and on the right, you have 100 terrorists on the Grand Place of Brussels. We have now enough material to use and to compare. And we also compared it, I don't show all the results here, for respiration. It was increased. And then we focused directly on the mitochondria because the main theory here was that higher oxidative activity was linked to metastasis. These are electron microscopy pictures. On the left, you have the poorly aggressive cancer cells. And on the right, you have the cells that we selected in mice. And these big formations here, black, they are mitochondria. So on the right, you see there are giant mitochondria compared to the left, where there are really small mitochondria. And the scale is the same between the 2 pictures on the left and on the right. So these mitochondria were enlarged, were really giant. And so they probably functioned in an aberrant way, which I summarize here. Our finding is I will just summarize here and not enter into too many details because I want to show you the clinical application that was published recently. So what we found actually was that in a, let's say, normal cancer cell, poorly aggressive one, you have the TCA cycle, you know, where pyruvate is processed to produce electron donors such as NADH and succinate. And these electron donors, they will donate the electrons to the electron transport chain, either to complex 1 or to complex 2. And then, if you remember your biochemistry courses, these electrons, they will travel from 1 to 2 to 3 to 4. And this will create an electrochemical gradient that will create a proton motor force, because then the protons will leave the matrix of the mitochondria and will start to accumulate in between the inner and the outer mitochondrial membrane. So this will create a concentration gradient, and the protons, all they want to do is to go back to the matrix. And they can go back to the matrix, but only through the ATP synthase. And when they go through to the matrix back using the ATP synthase, they release energy, and this energy is used to make ATP. Okay, so here we have a cell that is functioning normally, oxidatively, and which is producing ATP. What we found, so the phenotype we identified in the super metastatic and the super invasive cancer cells, is that there is an overload of the TCA cycle. It means that too many electron donors are produced, and they cannot be all accommodated by the electron transport chain. So the electron transport chain will be oversaturated. And if the electron transport chain is oversaturated, you have 2 things. First, the electron transfer is optimal. So the proton motor force is optimal, and the energy production is optimal. So this is perfect for a cancer cell. And the second consequence is that electrons will leak. They will leak out at different levels in the electron transport chain. And when you have that, so electron leak at a location that is not the normal one to react with oxygen. So it will react with oxygen, but with a single reaction to produce superoxide. So instead of water in a normal functioning mitochondria. What does it mean? It means that somehow the mitochondrion is a metabolic sensor that will produce superoxide, and superoxide could be used as a signaling agent to tell to the cell body you have to move because the environment is hostile. Okay, so this is the next hypothesis. ROS generating mitochondria can regulate cancer metastasis. And there was another study at the time where we were studying that in 2008, where Japanese people, it was published in Science, they engineered damages to mitochondria. So they didn't use the Darwinian selection that we used, but they experimentally induced defects in mitochondria. And when these defects were associated to the production of superoxide, if they transferred mitochondria from non-metastatic from metastatic to non-metastatic cells, they also transfer simultaneously the metastatic phenotype. So it means that the metastatic information is contained in mitochondria that are producing superoxide. So this was their finding, and it helped us a lot. Helped us a lot because we are in the same situation here. So we also used their approach, which we adapted a little bit to our models, but the philosophy is the same. So here we treated the cells with a mutagenic agent, and mitochondrial DNA is less protected than nuclear DNA. So at low dose, long time treatment, we will induce selectively a damage to the mitochondria. So this is the cells with damaged mitochondria. They are called row 0. And out of the row 0 total population, we isolated a clone, which is called D2. So if you look at the respiration rate of the cancer cells measured using oximetry, between wild types and row 0, you have decreasing by half of cell respiration rate. And then between row 0 and D2, you have a full block of respiration. The cells try to compensate by increasing the glucose to lactate conversion. So they increase the glucose uptake, and they increase the lactate uptake to try to rescue energy production. But at the metastatic level, for migration, for example, or invasion as another example, when you damage the mitochondria, but the mitochondria is still functional, migration and invasion are increased. When you fully damage the mitochondria, so let's say you delete the mitochondria, then migration is not occurring anymore, and it's even lower than before. Is superoxide responsible of this increase in migration? The answer is yes, because either when using a general antioxidant like epsilon, so it's an analog, well, its activity is analog to glutathione, which is the main antioxidant in a cell, you can suppress the gain in migration. And very interestingly for us, this drug, MitoTempo, is an antioxidant that is selectively targeted to the mitochondria. So it's an antioxidant fused to a triphenylphosphonium moiety, and the triphenylphosphonium moiety will accumulate where there is a mitochondrial potential, so where there is a negative charge, it means in the mitochondria. And so this antioxidant is rather selective, quite selective, let's say, of mitochondria. And so when you inactivate ROS, reactive oxygen species, in mitochondria, you block the gain in migration. We can refine it, so using specific inhibitors of the electron transport chain complexes. Here I show you the examples with rotenone, so it's an inhibitor of complex 1. The idea here is that we want to create a bottleneck. So we want to increase the leak of the electrons at complex 1 and at complex 1 selectively here with rotenone. So okay, if you increase the dose of rotenone, you will decrease by half at 10 nanomolar cell respiration. And if you increase the dose of rotenone, you will end in a situation where respiration is fully blocked. And so as long as the mitochondria is alive, but has a defect, migration will increase, And then when the mitochondria are completely inactivated, migration will decrease. So here again, we have a gain in migration when mitochondria are producing superoxide, which was measured using MitoSox. It's a fluorescent dye that is also addressed to the mitochondria with the same moiety, triphenylphosphonium, and so we can measure mitochondrial superoxide production. Again, this can be countered with N-acetylcysteine, a general antioxidant, epsilon, a general antioxidant, and mitotempo, a mitochondria selective antioxidant. And you can, research can be funny because you can complete the scheme with inhibiting complex 2 or 3 or 4, so creating bottlenecks a little bit everywhere, using different approaches like pharmacological inhibition, genetic inhibition, partial genetic inhibition, etc. And in all the cases, we found that we were able to increase cell migration as long as the mitochondria were still present and alive. In all the cases, and in all the cases as well, it was possible to decrease the gain in migration with antioxidants. Epsilon here, mitotempo here, and acetylcysteine. So giving rise to the giving this information here, which is what is the difference between the poorly metastatic and the highly metastatic cancer set? So again, the single terrorist, which is not very aggressive, and the large population of terrorists. Well, the difference is that there can be two differences. First one is an overload of the TCA cycle. Too many electrons are transferred, and so they will leak, simply because the electron transport chain cannot accommodate so many electrons. And this will lead to the production of superoxide. Another possibility is that there is a mitochondrial defect. And so in that case, electrons will also leak, for example, because there is a bottleneck somewhere. And so electrons upstream will just leak out. And they will also produce superoxide in the mitochondria. With the mitochondrial scavengers like mitotempo, we can block superoxide directly. And the superoxide derived H2O2, for example. And then we found this whole pathway here, which starts within the mitochondria with the activation of SARC. And then SARC is going outside of the mitochondria, and it will activate a cascade of events that will lead to the activation of a focal adhesion kinase. And these enzymes, they're responsible for the, let's say, remodeling of the cell cytoskeleton, which is absolutely necessary for migration and invasion. So we are here, as was told during the introduction of my talk, we are a lab of translational research. It means that we want to translate in the clinics or in applied science what we found here in fundamental science. And what does it mean with this scheme here? It means that we want to have the best possibility to identify the best target to prevent metastasis. What is the best target? Well, we know that upstream, we have at least two phenomena that can account for increased metastasis, and maybe there are 10 of them. Downstream of superoxide, we have here identified a pro-metastatic pathway. It's one, but maybe one among 1,000. So if we target one of these phenotypes here, some cells could escape. And if we target one of these downstream events here, cells could also escape by activating alternative pathways. And so we decided to target selectively superoxide. So this was done, as I told you, with mitotempo. But mitotempo is in the public domain, so it means that the drug itself will never make a medicament in the clinics, simply because nobody wants to pay for clinical trials with a drug that can be copied and sold by anybody. And so mitotempo, as was shown, can prevent the migration of the zero cells, but it can also prevent the migration of the super invasive cells that we had selected. And very interestingly, it can also prevent the migration of the super metastatic cells, the melanoma cells that we had selected. This is just an example. So here we injected cancer cells in the tail of a mouse. And so if you do that, they will generate metastasis, especially in the lungs, here, the top line. If you create a bottleneck in the electron transport chain, by targeting complex 1, we increase quite dramatically the number of metastases that are formed. So it means we increase the amount of metastatic progenitor cells. And if we try to counter that with MitoTempo, so by suppressing mitochondrial superoxide selectively, then we can decrease quite importantly the number of metastases. Remember that this is a model of super metastatic cells in a mouse that have been, that have been chosen because they are very, very, very highly metastatic. We still continue with MitoTempo. This is the model that we selected after 4 rounds of selection. On the left, you have the placebo-treated mice, number of lung metastases. And on the right, you have the mice that have been treated with MitoTempo, 0.7 milligram per kilo per day. If you look carefully at the single tumors, with the MitoTempo condition, most of the mice have no metastasis anymore. And the melanoma was injected under the skin of the mice. So it means that we are able to repress the full metastatic process, starting from the primary tumor to the end, and in the high majority of mice. And this type of cancer is an artifact from the lab, because this is the one that we selected to be super metastatic. So this is the worst case scenario, and we are very active with MitoTempo. We have very interesting results. A more, let's say, a more clinical scenario would be to have human breast cancer cells, to inject them in an immunodeficient mouse, so it will generate a breast cancer, and then we can treat the mice the same way with MitoTempo. So here we chose very aggressive, it's a triple negative human breast cancer that was injected in mice. We just modified a little bit the cells so they express luciferase, and so they become bioluminescent when they receive luciferin. So we can see them in the lungs of the mice here. And when mice were treated with the placebo, there were a lot of metastases in the lungs. And when the mice were treated with MitoTempo, no mice developed a single metastasis. And this is the result of 2 different and separate experiments, which was very encouraging for us. So if I have to make an intermediate summary here, we have a situation where ROS levels are very low. So this is involved in homeostasis. In the very opposite situation, if ROS production is very high by cells, we are in a pathological situation like Parkinson's disease, Alzheimer's disease. Cells get damaged and they will die. And if we are at an intermediate level of ROS production here, in the cancer case, we induce metastasis. So there is an increase in superoxide production, which is high enough to promote metastasis and low enough not to kill the cancer cells. In the case of Parkinson's and Parkinson's disease, Alzheimer's disease, hepatitis, a company based in New Zealand has developed a drug which they own through a patent, which is called MitoQ. And so here you have ubiquinone moiety, then you have a linker, and you have the mitochondria targeting triphenylphosphonium moiety. So this drug is very, very interesting because first, it will be targeted to the mitochondria. Second, we know that it will act in the mitochondria because of the charge. And third, we know that it will inactivate superoxide, but it will inactivate superoxide as a catalyst, similarly to a catalyst. So the drug here will not be consumed during the process. It will oscillate between ubiquinone and an ubiquinol form and use the electrons of the electron transport chain to be recycled. Now we are in the clinical application. We start with cells. What happens when we treat? So I will show you all the cell line together. We have very negative, triple negative breast cancer. You have a very highly aggressive human HER2 positive breast cancer. These cancers, when women are diagnosed with these type of cancers, one can predict that half of them will make metastasis, whatever the treatment done in the clinics. So this is a bad situation to have such type of cancer for half of the women. When we use MitoQ, we repress the cell respiration, which is high, which is a characteristic of the metastatic progenitor cells that we found. We decrease the mitochondrial potential, and we decrease the mitochondrial superoxide production, which was measured here using electron paramagnetic resonance with superoxide dismutase as a control. And so it means that this is the most specific way to measure superoxide itself within mitochondria. And if we look now at what happens to a non-cancer mammalian epithelial cell, you see that there is an effect on the cell respiration, but absolutely no change in the mitochondrial potential, meaning that the mitochondria will stay alive and fit, and no change in the production of mitochondrial superoxide, which was already low in these normal cells. So we have selectivity for cancer cells, and especially for pro-metastatic cancer cells, by using a mitochondria-targeted antioxidant. And then you have the consequences on the phenotype. So this is migration, so the percentage of migrating cells over time. The placebo situation is on the top, open circles, and then you have the mitotempo condition, where MitoQ condition, where you can see that the migration rate is decreased. We have the same for invasion. You see that invasion is strongly decreased when the cells are treated with MitoQ. And I have to say that the dose that we use here, 100 nanomolar, is the dose that we can reach in the plasma of human beings, actually the dose that was reached in the plasma of human beings in the phase 1 clinical trials. So this dose means something from a clinical point of view. And doses in tissues can be higher, because as I told you, the drug can accumulate within mitochondria, where it will not be consumed, but it will catalyze the deactivation of superoxide. For the clonogenicity, I think that the results are very clear as well. So this is clonogenicity on 2D, but we can also make it on 3D over time. Over the dose of MitoQ. And so the higher the dose of MitoQ, the lower the numbers of colonies. Cancer cells can also form spheres, and these spheres, they can be seen, as a model, as an in vitro model of a growing metastasis, of a growing secondary tumor. As you see, when we treat the cells with MitoQ, after a few days, the sphere is completely disorganized, and these cells here that are seemingly leaking out or escaping from the sphere, they're just dying cells, necrotic cells. Okay, let's go for therapy. We here used the simplest model, pre-treatment of the cells with MitoQ, followed by a tilvein injection of these triple-negative human breast cancer cells. Then we waited for 4 weeks. We biopsied the lungs, and they were insufflated and stained with Indian ink. And so with that model, we can see where the metastases are located, because as I told you, there is a problem with the perfusion of metastases, same as in the primary tumor. And so the white dots on the surface of the lungs, each dot is a metastasis. It's poorly perfused, so it will poorly take the Indian ink compared to the lung itself. On the left, you have the placebo situation, and on the right, you have the MitoQ treated animals. And I think that the pictures that are really representative of what happens in the whole group of mice that were treated, I think they speak by themselves. So here again, the very, very vast majority of the animals had no metastasis at all. There were some who escaped, maybe because MitoQ was not delivered the proper way. We don't know. Maybe because some tumor cells can resist the treatment, this can happen, and this is something we want to study. Why these escaped? But compared to the placebo, I think there are really few. And if we think in terms of mice that were, metastasis free after 4 weeks, we are in a winning situation. Even closer to the clinics is to inject the cancer cells directly in the mammary fat pad. Then we left a few days so that the cancer cells can take without any interference. And then the mice received MitoQ, which by chance, and I think it's a chance for the patients for the future if the drug is really active in patients. This drug can be delivered by oral gavage, so per os. We waited for 4 weeks, after which we had to remove the primary tumor, so the primary breast cancer, as is done in the clinics actually. The major treatment for breast cancer is to remove it surgically. And then we waited for 2 weeks and we did the same. We collected the lungs, insufflated them, and stained either with Indian ink or embedded for immunohistochemistry. And to a very big surprise, so this is the measurement of the tumor volume over the time. So you have the growth of the tumor in the vehicle-treated mice, and in the MitoQ treated mice, there was no statistical difference. So MitoQ is not active on the primary tumor. It did not prevent the growth of the primary tumor. But we were very big surprised when the primary tumors were removed. Tumors recurred in 100% of the animals in the placebo group, and in a very minority, 15% in the MitoQ treated group. What is the main characteristic of a cancer cell that can create a recurrence? Stemless. What is a main characteristic of a cancer cell that can make a metastasis? Stemless. And so we believe, and we are exploring that for the moment, that mitochondrial superoxide will induce stemness in cancer cells, and that we can repress it with a very high level of repression with mitochondrial targeted antioxidant, especially those that are specific for mitochondrial superoxide. So this is a Kaplan-Meier graph to show the number of recurrence-free mice over the time. And so each time that we go down, mice have recurrences. So this is the placebo group, all the mice got it. And here this is the MitoQ treated group where only a limited number of mice got recurrence. And when we analyze the lungs, the situation is even better than in the previous experiment, where with MitoQ we have an almost total repression of metastasis. Here we are, I think 80% of mice had no metastasis at all in the MitoQ treated group, compared to a number of metastases in the placebo. And again, this is the addition of different experiments on the same graph. Okay, the very last experiment, because I think I will be out of time otherwise. We wanted to confirm all the data by using spontaneous cancers in mice. So some mice that are modified genetically, they will develop cancers, and especially breast cancers, naturally, let's say, at a given age. So actually, these mice, they have been modified to express a viral protein. So they are called MMT, VPYMT. They express a viral protein in the breast cells that will create a cancer over time. And so after the birth, if you wait for about 6 weeks, tumors will be palpable in the breast of mice. And we decided to keep them untreated for about 4 weeks, exactly 4 weeks, sorry, to mimic a little bit what is done in the clinics. So on the day you diagnose a cancer in the clinics, this is not the day of treatment. Before, I mean, in between the day of diagnosis and the first day of treatment, there is usually a lag of time, 2-3 weeks, which is due to the fact that we have to book a room, to prepare the treatment. If there is a radiotherapy, there is a need to prepare a mask for radiotherapy, etc. So there is a lag time. So we kept the lag time as in the clinics, and then we treated the mice with chemotherapy, a combination of different drugs, as in the clinics in some protocols for breast cancer. And mice received, so they all received chemotherapy and plus minus MitoQ. And then we waited for 6 weeks and did the same, isolated lungs and looked at the metastasis. So as you can see, when we look at the primary tumor, so the weight of the primary tumor, there is no difference. Again, MitoQ is not active on primary cancers, but it significantly prevented metastasis, with here again, only 2 mice on the placebo group had no metastasis, and about, I don't know the numbers, I don't remember exactly the numbers, but a majority had less than 1 metastasis, 1 or 2 metastases in this particular model, which is a little bit further from the clinics than the previous model of human breast cancer. A typical picture of the lungs. This is the one that was taken by for the press release about the discovery. So again, each dot is a metastasis. This is the without, so the placebo group and the MitoQ group. If the lungs are black, this is because of Indian ink. And I'm at the conclusion to say that yes, indeed, the initial hypothesis is true. Tumor metastasis is under metabolic control. Second, both mitochondrial overload and mild respiration dysfunction can promote metastasis. And maybe other defects will also promote metastasis at the level of mitochondria, as long as they will be associated with an increased production of superoxide. Mitochondrial superoxide promotes tumor cell migration, invasion, and clonogenicity. This is demonstrated by the fact that very selective antioxidants targeting superoxide within mitochondria can block the whole process, all invasion, migration, invasion, clonogenicity. So if you use these kind of drugs, you can prevent spontaneous metastasis in experimental models or in the transgenic mice. And finally, MitoQ is, to me, a very promising drug to prevent breast cancer recurrence and metastasis. Why? For the two main reasons. The first main, as I told you, this drug, compared to others, is not consumed during when exercising. It's when exerting its antioxidant activity. And second one, it already successfully passed clinical trials. It is, but it belongs to a company which, when you are in the field of research, you realize it is an advantage, and because this company could perhaps pay for the clinical trials to test this drug in the future. I already showed you the graph on the right. This is just to remind you, too high levels of ROS can kill cells, in between promote metastasis, and actually they also promote resistance to chemotherapy and to radiotherapy, and very low levels are involved in homeostasis. And if you want to find more information about the second part of the story, you can go to the journal Cancers. So these are the two papers that have been published on the same date. There was a 1-day delay in between the two, but you have all the experiments and all the explanations there, and this is freely accessible. I just have to thank, of course, the people who contributed. So the main part of the work was done here at UC Louvain in Brussels with two main teams, my team and the team of Bernard Gallet, for the measurements of mitochondrial superoxide. University of Cambridge, and especially Christian Frezza and Michael Murphy, helped for a lot of analysis. Michael Murphy is the inventor of MitoQ, and the University of Namur helped with the electron microscopy pictures. The lead of the first part of the project, so the fundamental research and the discovery of the role of mitochondrial superoxide, is Paolo Porporato. He's now assistant professor at the University of Torino in Italy. And I have to acknowledge also Thibaut, who is the main technician involved in the project, and Valérie, a PhD student that was also involved in the project, fundamental research. And for the second part, it's Tania Capelloa here, who is postdoc currently in my lab. And so she's the one slowly converting MitoQ to a clinical treatment. And I hope that I will be able to keep her until the end of the way in a few years. So I was a bit long, but I thank you for your attention, and I'm open to receive any questions from your sides.
Thank you very much indeed, Pierre. There's a lot of information to assimilate very quickly, but it certainly is extremely impressive piece of work, and with very promising prospects for clinical application. We can now open to questions. If anybody wants to start asking questions, I come in just obviously. ROS is something which interests me a lot, because we have been over the years involved in iron metabolism. Iron is one of the catalytic ways in which you get to the really reactive ROS, which is not superoxide or hydrogen peroxide.
Yeah, the Fenton reaction. Right?
Yeah, but yeah, well, that's an interesting. This would also, is there any possibility that you might also be able to use a combination of MitoQ with metal chelation?
Yes, to the extent that we would be selective of the cancer cells.
Yeah.
Because you always have to remind that whatever you do to cells, cancer cells will find a better and a faster solution compared to the cells that are non-malignant. And so, to answer with some selectivity to your question, in the cytochrome C, in the electron transport chain, you have iron moieties that are indeed involved in the generation of superoxide, and that could indeed be scavenged or chelated. But then the question is, how selective would it be for the cancer cells?
Well, and it wouldn't help you because cytochrome C is the terminal link between complex 4 and oxygen. you need to knock out the major partner involved in getting electrons from complex 3 to complex 4.
Yep.
Sorry, we're now talking.
And usually when you activate the Fenton reaction, usually it goes from 0 to very high levels of ROS. It's very, it's a very proficient way to generate ROS. And there is a form of cell death which has been identified quite recently, which is called ferroptosis.
Yes, ferroptosis is now a big thing in the cancer field.
Yeah, and this ferroptosis, people try to exploit this in cancer, and here the idea is to promote actually the Fenton reaction, and so you will induce levels that override any redox defenses and kill cancer cells. And probably this ferroptosis is also involved in neurodegenerative pathologies, as I told, like Parkinson and Alzheimer's.
And
heavily
involved in those as well,
Yeah.
Question? You go ahead, Fabio. You've got a question. No, go ahead. We got somebody else with a question? Fabio's come up with a question here, which I think is interesting, but I'm sure you haven't got. It's a little bit early for this. We wondered whether it's just good for breast cancer or it might be good for other cancers and other, most cancers and metastatic.
So the answer is yes. It will be published soon. We have we have very similar results in prostate cancer.
Ooh.
And we decided to address prostate cancer first because, it's a rare type of cancer. Usually it's not very well addressed by industries because the market is small. But it can also, let's say, recur quite well and metastasize quite well. So the answer is yes, the same strategy works for metastatic pancreatic cancer. And in the pipeline, not yet started, but our next goal will be prostate cancer. And here the idea, sorry, here the situation is that when prostate cancers are overdiagnosed, and so after diagnosis there is no treatment for years, but only when the first metastasis occur, then they will be treated. And the idea here would be to deliver MitoQ from the day of diagnosis to the end of the life of the patient to prevent the entry in the metastatic phase.
Yeah, that would be very interesting. Yeah, very interesting indeed. Have we other questions? Looking around here. Yes, Ari. Ari, please. Ari Koskinen.
Thank you, Pierre. A very, very enlightening talk to an organic chemist like me. A little bit of background in medicinal chemistry also. Along the lines you implied also, one of the culprits to me seems to be the vascularization also. Can you comment on that?
Yes, so there are two ways drugs can reach a tumor. The first one is according to the concentration gradient, and in that case, let's say with time and the fact that the drug is administered repeatedly, with the time there is little influence of the perfusion on the targeting of the drug to the desired compartment. And then there is a second way, which is used by drugs that have a larger size, such as antibodies, which is convection. And so they will just follow fluid motions. And in that case, there is a high influence depending on the fluid motion and on the affinity of the, let's say, antibody, but it can be a protein, a nanobody, whatever else, and the receptor or the target. And so these two types of influence are different. They are less and more influenced by the perfusion. I think the answer is in the repetition of the administration of the drug. I have also to say that the organization of the tumor, these are very interesting experiments that were done years ago, but the hypoxic areas, they fluctuate with days and probably with weeks and cyclically. And so you have cycles of hypoxia oxygenation, And they are linked to modifications not only in the diameter and, let's say, functionality of the blood vessels, they can dilate and constrict, but they are also linked to the change in the structure of the blood vasculature, which changes really fast. So in 1 week, you can have a region that was totally hypoxic is totally oxygenated, and then the vessels will degenerate, and the week after it is hypoxic again. And so yes, so the answer is the same as for chemotherapy if you fractionate the dose. So if you deliver every day a little bit, then you reach your objective, and the limitation in perfusion has a limited impact on the treatment. In the particular case here of MitoQ, if we reach the cells that are already in the mainstream, in the bloodstream, in the main circulation, we reach our objective. And this was demonstrated by the experiments where we injected the cells in the tail vein of the mice. And they were pre-treated with MitoQ. And so even if they escape from the primary tumor, they reach the blood, and they're on their way to make the metastasis, we can cut down, shut off their stemness capacity in the blood before they stop in the organ of destination. And so here, of course, the perfusion has no impact anymore.
Excellent.
Thank you very much. That's helped a lot. Thank you.
Welcome.
Any more questions? No? No? Yeah? Bruno? Hello, Bruno.
Yeah. Hello. I'm the other organic chemist in the audience. So thank you very much, Pierre, for this very interesting talk, really. So am I correct that the drug has to be in the blood, in the bloodstream?
Well, the drug has to accumulate in the mitochondria, and we know it accumulates at the inner membrane of the mitochondria, and especially it accumulates between the complex 1, 2, and the coenzyme Q10, which is the shuttle that naturally transfers the electrons between 1 and 2 and complex 3. And it will, in fact, it will compete with the natural coenzyme Q10, which is located there. And the fact is that MitoQ can take the electrons from complex 1 and 2, but it cannot transfer the electrons to complex 3. And so the exact mode of function of MitoQ is in between deactivating superoxide that has already been produced. So this is one possibility. And because it cycles between oxidized and reduced forms, it will catalyze the reduction down to water, and not only down to H2O2, which is great because we believe that H2O2 is an intermediate in the signaling, because it has a longer half-life than superoxide, of course. So this could be one mode of activity, and the second mode could be it will take the electrons that leak out, or it will even force the electrons to leak out of the respiration of the electron transport chain. And if you go to high doses of MitoQ, which are well above what can be reached in a human body, and we have done these experiments, you can switch off respiration completely.
But is it administered orally or
orally? Orally.
It's orally. All right.
Yeah.
And could you re-explain the function of the positive charge on the phosphorus?
Yeah. So this will. I mean.
So why is it necessary to have a plus charged?
So the inner mitochondrial membrane is charged negatively because the protons go from the matrix to the intermembrane space, and so it creates a negative charge on the inner part of the inner membrane, and this is exactly where MitoQ will settle. So plus in the triphenylphosphonium, the positive charge will be attracted by the negative charge at the inner side of the inner mitochondrial membrane, where the drug will accumulate actually. And the fact it is a triphenylphosphonium, it will allow the drug to stay as membrane permeable. So it's lipophilic enough to permeate the membranes and charged enough to accumulate with the negative charges.
Yes, we do research with lipophilicity, and so I wasn't actually aware that charged drugs like this existed. So that's quite interesting. But
yeah.
the plus charge must have a counter ion, a negative ion, and
yes.
I had a quick look in publications, and it's always presented, it's always pictured without the counter ion.
Yeah, I don't have it in mind. I think it's mesylate or something like that for MitoQ. I am not totally sure. It's in the paper, of course, but I think that the counter ion gets stuck outside cells. It will not enter, and the transport is not via transporter, so it's just permeation, membrane permeation. So I think the counter ion stays stuck outside of the mitochondria, and only the positively charged.
Actually, I don't think so. I think it is the two ions shield the charge, and that's how it goes through the membrane.
But I don't know. I'm not a chemist, so you know better than me.
I'm gonna have a look at that. Yeah.
And there are many drugs like that because they made no mito vitamin C, mito vitamin E, mito. This is declined in many. So people try to address anything to mitochondria using this moiety, and apparently it works when you look at the literature. People get a selective effect on mitochondria when using this way to target mitochondria.
Yeah, very interesting.
And what is interesting by the cancer cells is that the mitochondrial potential is higher compared to the non-cancer cells. And this is part of the selectivity. As I told you, the metastatic progenitor cells are very oxidative compared to the regular cancer cells that are already more oxidative compared to the normal cells. I mean, part of them.
Yeah.
And so this is if stemness and high respiration rate goes together, then it means that the high mitochondrial potential will go together, and it means that mitochondria will target preferentially these cells compared to any other.
Yeah. Okay, very good, interesting. Thank you.
Thank you very much. Thank you. Other questions? No? Okay, then we'd like to thank you once again. It's interesting, I think, philosophically, that we know that low levels of reactive oxygen species are used for signaling, so they're important. We also know that high levels of reactive oxygen species are toxic because they kill cells. That would be the ferroptosis story. But we now have a very interesting situation that the no man's land in between a little and a lot is just as dangerous if you happen to have a tumor.
Yes.
That would be the take-home message, I think, I'm most impressed by. I hadn't realized that you could have an intermediate level of oxidative stress that could be potentially more dangerous in a tumor situation.
And the big question, and this is for the chemist, the big question is, can we have an absolute value? Because for the moment we have only relative values, and we don't know the exact amount in nanomolar or micromolar or whatever of mitochondrial superoxide. I think there are ways with EPR and MR, but it would be great to know exactly, because then we could also work on the stoichiometry between the antioxidant and the superoxide.
And essentially titrate the patient to the level you want.
We need to make a titration curve in a patient or in a mouse for mitochondrial superoxide in particular. But this has to be done. It was not done to my knowledge.
Well, thank you very much again. I think you've given us a lot of things to think about, and a lot of people who've, like myself, had to do with cancer. It's very nice to think that there might be some kind of positive way of dealing with metastasis, which is the bane of the cancerologist's life, to be honest. I mean, you said it rather bluntly, and it is rather true. Once you've got a number of metastases in several tissues, it's palliative treatment. So, that is a very important point. Thank you very much. We've enjoyed it.
And again, it was an honor for me to be at the Royal Society of Chemistry. Thank you very much.
Thank you, Pierre.
Have a nice evening.
All of you, thank you for joining us, and we will see you next season. We look forward to seeing you next season. The first lecture of the year will be on the 29th of September, Vincent Lemaitre, and I trust, we hope, that this will be live, but you will find more on the site of the Royal Society of Chemistry Belgium section. Thank you once again, and have a very nice evening. Bye-bye.
Thank you. You too. Bye-bye.
Bye.